Okay, so hi guys, good day. This is Dicot Medical, and today we're going to be talking about the cell. So guys, this is our outline for today on cell. First, we're going to define cell and explain it. Then we're going to talk about the general structure of cell. We're going to talk about the organelles of the cell, membrane-bound and non-membrane-bound organelles. Then we're going to talk about transport across cell membrane. There's passive, active, vesicular transport. And then we're going to wrap everything up and tie everything together by talking about you as a cell. Okay, so the first thing we're going to be talking about today is going to be the cell as the structural and functional unit of life. Okay, I'm very sure most of us, if not all of us, are very, very familiar with that definition of cell. So how about I go deep into that today? So firstly, the structural unit of life. Okay, take yourself as a human being now. You are alive. Yes, we can see we have arms, I have a face, I have eyes, nose, and all of that. But then the structure of myself as a human being actually all boils down to cells. Let me explain. So your skin, my skin, is actually an organ. That's right, organ. And then if we take a slice of my skin, if we want to look at my skin under a microscope, cell slide preparation, we're going to need to take a slice of my skin, and that slice of my skin is going to be called a tissue. I'm going somewhere. Tissue. Now, if we zoom in on that tissue, when we place it under the microscope, if we view it at whatever magnification, we are going to see a bunch of cells. Okay, great. Now, let's take that into clear perspective, the small intestine. So, a cell, if you take a slice of a slice of the small intestine to view under the microscope, you're going to see a bunch of cells, and those cells are actually called simple columnar cells. Now, a tissue is a bunch or a group of similar cells, similarly shaped cells that serve the same function or that have the same function. So in the small intestine, the simple columnar cells there, the tissue is going to be a bunch or a collection of simple columnar cells that have the same function of absorption. That's the function of columnar cells. Columnar cells function in absorption. This is what they look like. So this is just a rough diagram. This is just a rough diagram. But they're tall. They're tall. Okay, and they have some processes. They have some processes called villi. Microvilli. Okay, so now, like I said, that's just a slice. Just a slice we take from the skin or from the small intestine. We put under the microscope and we can see they are cells. So now those cells, the group of them, same function, that's the tissue. Remember, we took the tissue from an organ, the small intestine, which is an organ, and then the small intestine is part of what? It is part of the gastrointestinal system. GIT, gastrointestinal system, GIT system. So this is true. This explanation I just gave now is true for various cells, various systems around the body. For example, now in the cardiovascular system, which is made up of the heart and the blood vessels, the cells lining the blood vessels, endothelial cells, simple squamous. The cells of the heart, which is an organ, if you take it to part of the tissue, the cells of the heart are called cardiomyocytes. Okay, so that is cell as a structural unit of life. Like it provides the actual structure, even for bones. Even for bone, if you do slide preparation for bones, a slice of bone, you will see cells. So you can imagine yourself as one big cell. Although, since cells they're so small they can't be seen with the naked eye. We have to go under different magnifications with the microscope, but they still comprise the whole body. So in cognizance, in mathematics, whatever you want to call it, we actually have about 100 trillion cells. Okay, so you're just a big bag of cells. You just imagine yourself as one big cell which contains about 100 trillion cells. Okay, so that's cell as structural unit, and a cell as the functional unit. Now, back to our explanation on the gastrointestinal system. The function of the gastrointestinal system is for digestion and absorption of food. That's a major function. There's going to be There's going to be mechanical digestion, there's going to be chemical digestion, there's going to be absorption, and then there's also going to be passing out or excretion of waste. But let's just focus on the digestion and absorption of food. So these simple columnar cells now that I told you that are in the small intestine, they function in absorption. I said they function in absorption, and they're able to do this. Why? Because of the microvilli. Now, if we go a bit, if we go closer, or if we increase magnification of the columnar cells, of the small intestine, they're going to look like this. Okay. So now this is microvilli, and then what does microvilli do? So microvilli, they're actually foldings of the cell membrane, and they work to increase the surface area. Okay, they work to increase the surface area. Elementary knowledge from physics, chemistry. Increase the surface area facilitates diffusion. Factors that affect the rate of diffusion. Keep that in mind because we're still going to talk about it today. So basically that's the function of, and so this simple columnar cells, they are called enterocytes. Simple columnar cells of the small intestine are called enterocytes. So cell being the functional unit of life, like I said, majorly the major function of the gastrointestinal system is for digestion and absorption. Function, absorption. They are the ones that function in absorption. They are lining the entire small intestine. So when food or chyme gets into the small intestine, they absorb water, they absorb electrolytes and nutrients and all of that. So that is how cell is done. Now I've explained how cell is the structural unit of life, how cell is the functional unit of life. Now let's talk about how the characteristics of cells. So characteristics of cells, they are not difficult. They're very, very simple. So let's just say this is an elementary cell. That's the cell, this is the nucleus, and this is the cytoplasm. So number one characteristic of a cell is that it is able to make use of nutrients and turn it into energy. It would ingest, we will learn about that, we would ingest food particles or debris, or food particles, and then make use of the food particles. It can also ingest debris, yes, but in relation to energy, it can ingest food particles, make use of that through some of the organelles, and then produce energy for itself. That's number one. Number two, the cell can also get rid of, we would also talk about how we do that. The cell can also get rid of waste products from itself. These are characteristics of the cell. It is functional, like every single cell can do this. Okay, it can ingest and it can also evict. Okay, so now let's talk about the general structure of cells. So now I told you this is just an elementary diagram of a cell. So plasmalemma. So a cell, so there are three main parts of a cell. Number one is the plasmalemma, which is the cell membrane. This is the plasmalemma cell membrane. Number two is the cytoplasm. So now this space is the cytoplasm. So the cytoplasm of a cell contains cytosol. Cytosol is a fluid or gel-like substance that it is inhabiting the cytoplasm. Now, the function of cytoplasm is to hold organelles. So cytoplasm is where the organelles of a cell. The cell has many organelles, different organelles, which all have their function. So the function of cytoplasm is to house those organelles. And then the function of cytosol, which is found in cytoplasm, they are not one and the same exactly, but their function is very, very related. Their functions are related. So the function of that cytosol, it would balance those organelles. So it's like how you can, a bucket, if you have an ocean, you can put a bucket on the ocean and to balance it would float. So that is basically what happens. That's one of the functions of the cytosol, holding those organelles like that. Cytosol and cytoplasm, holding those organelles like that. And then number two, it gives a fluid medium for chemical reactions to take place in each of the organelles in a cell. Different reactions, different base reactions are taking place. So the cytosol gives fluids, like it gives a fluid medium for those reactions to take place. I hope you are getting me. Okay, so yep, so I've done. So now we are going to go to our organelles of the cell. Remember I said the organelles are found in the cytoplasm. Oh, sorry. The third very principal component of the cell. You know, I said the first one is the plasmalemma or cell membrane, second one is cytoplasm, and then the third principal component of the cell is the nucleus. The nucleus. We will talk about the nucleus. So now let's go into our organelles. So since we're talking about organelles, I have to introduce one thing to you before we go into organelles. So plasma membrane, plasmalemma. I am going to go deeper into the plasmalemma. I'm going to show you why this is important in relation to organelles, but let's just firstly explain the plasmalemma. So the plasmalemma is a phospholipid bilayer. It is the composition, the structure is phospholipid bilayer. So what does that mean? Phospholipid. This is a phospholipid. Like a phospholipid bilayer, that's anything, whenever you want to hear plasmalemma, whenever you hear cell membrane, that's the first thing that should come to your mind. So now this is a phospholipid. Phosphate and lipid. So that means it would have a phosphate group, this phosphate, and then a lipid group, this lipid. Now that's phospholipid. So the cell membrane itself is actually composing of different macromolecules. Each macromolecule composing the cell membrane has its specific function. So we have lipids, we have proteins, and we have carbohydrates. So protein actually has the highest content of the cell membrane. Just look at the diagram here. The protein has the highest content of the cell membrane, which is 45 to 54%, but just take it as 55%. The lipids has the second highest, or runner up. Just take it's 35. I wrote 35 to 40%, but you can also just take it as around like 20, around 49% or so, around 40, and then carbohydrates just 3%. So our lipids here, the lipids in our cell membrane are phospholipids, cholesterol, and glycolipids. Okay, phospholipids, cholesterol, and glycolipids. Obviously, as we can see here, phospholipids will have the higher content of all our lipids. Phospholipids will have the highest content, then followed by cholesterol, and followed by glycolipids. So now, this phospholipid layer, since it has a phosphate head and a lipid group, that means it can be hydrophobic and it can be hydrophilic. Hydrophobic, I know we all know what phobia is. Fear. Hydrophobic, understand it as I'm afraid of water. So our hydrophobic layer or hydrophobic part is the lipid. Elementary knowledge, we know that fats and oil, or fats or oils, don't mix with water. Oil and water, they don't mix. Fat and water, they don't mix. So just take it as that. Lipid is actually fats. You would learn that later, but lipid is fat. Lipid, you can, the name is interchangeable. Lipids are fats. So since fats and water don't mix, lipids and water don't mix, just take it as that. Lipid and water. So that's why the lipid side, this lipid part, is hydrophobic, and then the phosphate side is the one that is hydrophilic. I feel it, I like it, I want it, I can withstand it, or I can move with it. That's so just the part that accepts water, phosphate. The part that doesn't accept water, the lipid group. So if we come here now, this is the structure of the cell membrane. This is the molecular structure of the cell membrane. So we can see here that the lipid or hydrophobic tails, they are together here. They are like a gate. Just imagine this as a gate preventing water and water soluble materials from entering the cell through them. Okay, and then this phosphate group is just at the edge, like the roof, and then the ground or the base. Just imagine a house, the roof, the top, this place and this place. Those are the phosphate heads. Then here, the door, which is supposed to allow substances inside, this is the lipid layer, and it's what would prevent anything that is not lipid soluble. So that means that lipid soluble materials can pass through, and that will be explained. But example of lipid soluble materials, oxygen and carbon dioxide. So they can pass in and out of the cell membrane at will. So okay, now these things that I drew here, this here and this here, those are that represents cholesterol molecules. So cholesterol is also important in the cell membrane because cholesterol controls the rigidity of the cell membrane. increase in cholesterol levels or increase in cholesterol in cell membrane reduces the fluidity of the membrane. So fluidity, so okay, if the cell membrane has a fluid mosaic model, it is able to, you know, change shape. It's fluid in nature. But increase in cholesterol, you know, just imagine like a rubber band. Just imagine a rubber band. Increase in cholesterol would increase the rigidity and reduce the fluidity of the membrane. I know it doesn't work, but let's just imagine one could freeze dry or freeze a rubber band. That's what cholesterol does to the cell membrane. It takes away the elasticity, just keeps it rigid like that. Okay, so and then so glycolipids is just glucose molecule or glucose moiety attached to a lipid. You know, most of the things in medicine, most definition, most words, it's actually just a combination of two. And if you can understand those two elementary ones, put them together, you're very good to go. Okay, so that has to do with the lipid content of a cell membrane. I've explained the phospholipids, cholesterol, and glycolipids, just glucose attached to the lipid. Then we have proteins. Okay, so our proteins here, they are of two types. Normally in a cell, proteins can either be structural or functional. Since this is here, as we can see, it is aiding in the structure of the cell membrane, which means it is structural protein. So now, part of the cell or the cell membrane proteins that we have, there are two types: integral proteins and peripheral proteins. Okay, let me just write this here. I'm going to clean some of this, so if you want, you can take a screenshot of this, or you can just save this video for later. Integral proteins and peripheral proteins. Okay, I'm very sure you are so smart, so maybe you already understand the gist of what's about to happen or what's going on with these proteins, but I'll just still explain it. So integral proteins, these big proteins, they are integral proteins. So these are the proteins I'm talking about. So these big proteins here, they are the integral proteins. They span the entire length, as you can see, the entire length of the cell membrane. They span it. Okay, and these those are integral, and then these peripheral proteins, they just attach. They don't span the entire length. They just stay on the cell surface. Okay, so these are peripheral proteins. So now, that's the structure. So now the function. Integral proteins, the major function of integral proteins is that they serve as channels. They serve as channels for ions. Okay, so sometimes ions enter the cell, ions leave the cell. So these integral proteins serve as channels for those ions and for all those other substances that cannot pass through our phospholipid bilayer. Ions, especially charged ions, they do not pass through the phospholipid bilayer. They need integral proteins to be able to pass. And then our peripheral proteins also serve arguably the same function of trying to transport molecules, but not in the same way. So since our integral proteins are this big, they serve as channels, they can open for it. But then these are our peripheral proteins, they're not so big, they don't span the entire length. So what do? They serve as carrier proteins. Remember that, carrier proteins, because we are going to go somewhere with this. This entire cell, everything about the cell is you're going to be built up on it throughout your entire stay in medical, throughout your life, and it can also apply to you in other areas of your life. But let's just get back to this. Okay, so our peripheral proteins help us as carrier proteins to carry across the cell membrane. Okay, so that's all for the proteins of the cell membrane. Then we're going to go to carbohydrates. So this moiety you can see here is actually the carbohydrate molecule that are attached to the protein. And what does that make it? That makes it a glycoprotein. Okay, so now, so that's the structure. Is attached. It can either be attached to a protein or it can be attached to a lipid. So now the function of the carbohydrate molecule is actually number one most important. Keep in your mind, if you want to gain anything from this part and what to talk about, keep in your mind. Number one is that carbohydrates have to do with, or they generate the negative charge of the cell membrane. So the action potential, or the potential, sorry, not action potential, the potential difference across the cell membrane, the inside of the cell is always going to be negative, and this owes to the carbohydrate moiety. Number one function is that it gives the cell the negative charge. The negative charge of the cell, we are owing it to the carbohydrate molecule of the cell membrane. That's number one. Okay, so no, let's just write negative charge. So, and then number two function of that carbohydrate molecule is that it acts as it acts for as a receptor for signaling. Cells, now according to this diagram that I put here, cells, they are not just in isolation, okay, they are beside each other, so it's like a cell passing or beside, or let's say like a hair hormones. Great wonderful example. Now a hormone comes in molecules looking for where to, the different cells can be surrounded by each other, one group of cells, so to be able to signal, hey, I'm here, bind to me, we use our carbohydrate molecule. We can also use our protein molecule, but for the function of carbohydrate, let's just base it as for carbohydrate, our carbohydrate molecule. Okay, I think that is all about our cell membrane. And then also to note about the cell membrane, now we're going to talk about the function, the actual. So this entire thing I've just finished talking about is the structure of the cell membrane. So now we'll talk about the function of the cell membrane. So number one, it encloses the cell. Okay, it gives the cell a structure. Okay, protects the cell. Number one, it gives the cell a structure by enclosing it. Okay. Number two, it protects the cell from external, external forces. It's protected as protection. Number three, it is like a guard, a gate. Selective permeability. Okay, there are some molecules, like I explained before, there are some molecules that cannot enter the cell directly through the phospholipid bilayer. They find their way either through the channels via the integral proteins or the peripheral proteins, or whatever way possible. There are different ways of transport across cell membrane, and if you stay to the end of this video, you will see them. Okay, so yes, so and it gives rigidity. It has so many functions, this cell membrane. Okay, just remember, number one, it encloses the cell. Number two, protection from outer. Number three, selective permeability. Okay, great. So now we're going to go to this structure. Don't be alarmed. This structure is actually a combination of about two, if you bend your eyes small, three different structures. So let's bring out the first and most important structure. Hold on. Before we do that, I said there was a reason I was talking about the cell membrane first before I went into organelles. So in a cell, organelles can either be membrane-bound organelles or non-membrane bound organelles. Why is this important? I told you, in each cell or in every cell, processes take place in those cells. So some processes that take place in certain organelles can be very destructive towards the atmosphere of the cell, the inner atmosphere of the cell. So to prevent that, attack on the cell, or to prevent destruction of the cell, some of those organelles need to be enclosed, okay, so that whatever is happening inside the organelle, whatever is causing degradation inside the organelle, will not leak out into the cell and destroy the cell. Okay, great. So now I'm going to talk about the membrane-bound organelles now. So number one we have on our list is the nucleus. If I bring it out, this is the nucleus, and it has something inside. It looks like a miniature cell, I know. So now the nucleus, this nucleus we have outside, it has a structure inside called a nucleolus. So the nucleus is surrounded by a nuclear envelope, and this nuclear envelope actually has an opening called nuclear pore, with which what is going on inside our nucleus is able to communicate with these structures outside, which would be explained. So our nucleus actually houses chromatin. So these strands here, they are chromatin, and from your biology, chromatin is what makes up chromosomes. But what makes up chromatin? Chromatin consists of DNA and histones. What is DNA? DNA is gene. It contains the genetic information of an individual. Histones, they are the proteins that are involved in the wrapping to constitute a chromosome. So our nucleus contains that. So that is why our nucleus contains genetic information, because of this chromatin. Okay, function of our nucleus. Our nucleus is very important in protein synthesis. Okay, protein synthesis. Then we go to our nucleolus, which is our structure inside our nucleus. So now these dots that I just finished putting here and these dots that are here, they're actually called ribosomes. Ribosomes are what the nucleolus produces. No, the nucleolus is not membrane bound. The nucleolus does not have a membrane surrounding it, but it produces ribosomes, and ribosomes are also important in protein synthesis. Okay, we've spoken about our nucleus. Now we're going to talk about these structures around our nucleus. So now, these structures, they are actually very similar, but from what you can see right now, this is dotted. Okay, it doesn't look so sweet, so nice, but this one is clear, it looks nice. So this dotted organelle is called the rough endoplasmic reticulum. Number one, these organelles are called endoplasmic reticulum. Endoplasmic reticulum, that's ER. Now, this part that is dotted is actually called the rough endoplasmic reticulum. And our rough endoplasmic reticulum is from what you can see right now, actually, it is just a folded overlapping layer of vesicles. Not of vesicles, sorry, but it's a folded overlapping layer. Okay, a network of tubules. Okay, and flat vesicular structures, but not vesicles, flat vesicular like vesicles structures. Okay, so the function of our rough endoplasmic reticulum, our rough ER, is protein synthesis. Okay, protein synthesis. These dots that are around it, they are ribosomes. So the ribosome that is produced in the nucleus, in the nucleolus, comes out. Remember, because nucleolus is not a membrane-bound organelle, it's not membrane-bound. So the ribosome produced here comes out into the nucleus, and then because of these nuclear pores, it is able to communicate and come into the rough endoplasmic reticulum. And what did I say ribosomes are for? Ribosomes are for protein synthesis. What is the function of rough endoplasmic reticulum? Protein synthesis. Okay, wonderful. So now, these structures here are also endoplasmic reticulum, but they're agranular, so they're called smooth endoplasmic reticulum. So now, the smooth endoplasmic reticulum has two functions. Number one, lipid synthesis, and number two, detoxification. So remember, rough endoplasmic reticulum is for protein synthesis, and number two, smooth endoplasmic reticulum is for lipid synthesis. Now, when protein is synthesized, we already have explained to you how that happens, in a small way, a brief way. When protein is synthesized, when lipids are synthesized, how do they now get to where they are needed? How do they get to to organelles or other places in the cell, or how do they get out of the cell? I'm going to introduce to you our brother, the Golgi body or the Golgi apparatus. So now, standard definition of the Golgi apparatus is that the Golgi apparatus is a 4 or more layer. Here I drew about 5. So it's a 4 or more layer of flat vesicle structures. Flat vesicle. So from what we can see now, with the way it's right now, see the things that are around it. Okay, these things are around it. They are just imagine it's budding off. These things, they are called vesicles. Okay, vesicles. They are budding off the Golgi body. The Golgi body is like a logistics company. Basically has buses or cars, sends out buses or cars. The function. So that's the structure of the Golgi. So these vesicles, just imagine them as baskets. Okay, they want to carry, just carrying everything. They also have another function, but because majorly for transport. So now the function of the Golgi body, majorly for transport. So as whatever is coming out of here, the protein, the lipid, this Golgi body will just go up, carry it and transport it. So it will carry it and transport it. So that Golgi body, so that makes Golgi body very, very prominent in secretory cells. For example, secretory cells that secrete, e.g., hormones or saliva. But cells that secrete hormones, like the thyroid gland secretes thyroid hormone, it would have the cells of the thyroid gland would have a high concentration of Golgi body. Why? Because it is a secretory cell. Do you get? Okay. So another function of the Golgi apparatus is that it buds off to give us another organelle. Follow me, I will show you the way up to the other organelle. So this is the other organelle that the Golgi apparatus, the Golgi apparatus or the Golgi body, it is only this organelle that it buds off to give, according to me. It is only this organelle. So this organelle here is called a lysosome, okay? Now, listen to me first. Don't look at what is written here. Just listen to me first. So these two, okay, they are two different organelles in the cell, but they have similar function. Two different organelles in the cell, but they have similar function. I also drew them the same way to show you that they actually have similar function, even though their mechanism of action is different. So the first one here is a lysosome. Now I put H2O here. Why? Wait first, I'll explain. So the lysosome, obviously, as you can see here, is a membrane-bound organelle, double lipid or bilipid layer. The membrane is also a bilipid layer. Same thing with the nuclear envelope, it is a bilipid layer. So now the lysosome, okay, inside it's a vesicle. Number one, it buds off. It is the lysosome that buds off the Golgi body, the Golgi apparatus, to become what it is. So inside the lysosomes, okay, we have the hydrolytic enzymes. What are hydrolytic enzymes? So these are enzymes that just help to degrade or break down debris or food via hydrolysis, which is via separation using a water molecule. That is why H2 is inside. So let's bring this out. So if I were to separate H2, it would be into hydrogen and hydroxyl ion. So how does hydrolysis happen? So let's use the example of a protein. Now I told you lysosome, apart from breaking down or apart from wanting to break down debris and damaged cellular parts, lysosome also functions in cellular digestion. So let's take protein for example. You just finished drinking milk and then you're sitting down comfortably. Now what goes on inside your cells? This is it. So now the protein, via some cellular transport mechanisms, is able to enter the cell. Time for digestion. This is what happens. So a protein is made up of amino acids. A protein is a bunch of amino acids joined together by polypeptide bond. So amino acid joined together. So what happens in hydrolysis hydrolytic enzymes are the ones that carry out hydrolysis. So what happens in hydrolysis is that these two ions that have been separated will now separate or destroy the peptide bond, destroy the bond between two molecules to separate them. So once hydrolysis takes place, or once hydrolysis has happened in this molecule, what's going to happen? A is going to be separated via hydrogen amino acid, and the other amino acid is going to be separated via hydroxyl ion. Okay? So hydrolysis, breakdown. Lysis or lysis means breakdown. Hydrolysis means breaking down by water molecules, using water molecules to split or to break down. So that's the function of lysosome. That's the structure and function of lysosome. So now the brother of lysosome, or the cousin, whatever you want to call it, is the peroxisome. Okay, the peroxisome also carries out the same or a similar function. Okay, they all have the same goal, but the peroxisome, number one difference is the peroxisome buds off the rough endoplasmic reticulum. The peroxisome buds off here, but the lysosome buds off here from the Golgi body. Okay, that's number one. And number two, the peroxisome contains, instead of hydrolytic enzymes, it contains oxidative enzymes. So let me tell you the mechanism of action of how the peroxisome works: to digest, protect from bacteria and other microbes that shouldn't be found in the cell, and to destroy damaged or dead organelles inside the cell. Okay, so inside a peroxisome, okay, we have oxygen, which is the oxygen that is here. Inside the peroxisome we have oxygen. And now around the cell via different organelles, different chemical reactions that are taking place. Hydrogen is going to be released. Hydrogen is going to be found in the cell due to these chemical reactions that are taking place around the cell, around the peroxisome. So via oxidative enzymes, the peroxisome is able to combine oxygen and the hydrogen gotten from outside to create hydrogen peroxide, H2O2, to create hydrogen. So the hydrogen from outside, the oxygen from inside is able to come together and create hydrogen peroxide. With this chemical reaction is stabilized or is maintained by oxidative enzymes. So now we have hydrogen peroxide. How is hydrogen peroxide able to perform these functions of digestion, breaking down, splitting, debris, destruction, etc.? Via catalysis. So catalase enzymes, they are what they work together. Catalase enzyme plus hydrogen peroxide is what is able to perform these functions. Okay, so next we are going to go to is this, the mitochondria. Yes, clap for yourself. I know that if you were not familiar with anything I've been saying since, you'll be very familiar with this mitochondria. So the mitochondria, as we can see, or as we already know, number one, we know of all these organelles I've been talking about, number one thing we know about them is that they are membrane bound. So mitochondria, number one, is membrane bound, is a membrane bound organelle. But the mitochondria is kind of peculiar. Do you know why? Because it's the membrane bound, or its membrane has an outer layer and an inner layer. And the inner layer of the mitochondria is thrown into folds. Okay, these loops. This is the inner layer, and the mitochondria, the inner mitochondria membrane is folded into these loops. Okay, and inside these loops is mitochondrial matrix. It's a fluid in the mitochondria, aiding in the chemical reactions of the mitochondria. So that's for the structure of the mitochondria. Then the function of this, mitochondria, this is the spelling. So the function of the mitochondria is the powerhouse of the cell. How is it the powerhouse of the cell? Mitochondria is what provides energy for the cell, for all cellular organelles to be able to work, all cellular processes to be able to move forward. We need energy. And what you need to know is that the mitochondria is the one that will pump out that energy. And that energy is in the form of ATP, adenosine triphosphate. Adenosine triphosphate. Okay, just bear that in mind. So that's for mitochondria. And then here, this is just a secretory vesicle. Okay, it is just, obviously it's membrane bound, and to contain whatever needs to be secreted, most times it's proteins. So as I said earlier, it's going to be majorly found in cells that function to secrete. Can be secreting mucus, can be secreting hormones, but they are secreting those cells that secrete, okay, or tissue that secretes, or an organ that secretes, would have concentration of secretory vesicles, concentration of Golgi apparatus or Golgi body. So now this structure here, it is the organelle I'm going to introduce to you today that is not membrane bound. It is called the microtubule. It is part of the cytoskeleton of the cell. The other two parts of the cytoskeleton of the cell are microfilament and let's leave that last one. Let's leave it. I'll come back to it. Okay. So the microtubule, the function of the microtubule is to provide rigidity for the cell membrane, structure. Think of it, it looks like a pipe, but think of it as logs of wood. Gives structure, rigidity to the cell. And then cilia. So cilia is a process. There's a process on the cell membrane. Process is coming out of the cell membrane, but it's a specialized process of a cell. Okay. So cilia or ciliated cells are majorly found in the respiratory, or lining the respiratory tract, in the respiratory epithelium. So this cilia beats, elementary knowledge of biology. So what it functions to do in the respiratory system is that when air is inhaled, your nasal cavity down to your your pharynx, your larynx, to your trachea, the bronchioles. No, down to the trachea to the bronchioles, they are lined with this respiratory epithelium, which beats, okay, the cilia beats, which functions to, so in the motion of the beating, it functions to take out or to bring out or to trap bacteria or debris like pollen grain, pollen dust or asbestos that gets trapped in the respiratory system, okay, to prevent it from causing irritation, etc. Okay, so if you were able to gain even one thing, or if you laughed at just one joke, please press the subscribe button or hit the subscribe button.
We are Dicot is fam. Please don't forget to like, subscribe, and forward this video to whoever needs it. Bye.